let's talk about mitochondria. Acidophilic structure loves acid. You're gonna see it pink from the mitochondria protein when we look into the light microscope stained with hematoxin eosin. And this guy is very strange organelle. It's a little bit autonomous. Some people call it semi, meaning half autonomous. And I will explain in a minute what that means. And it is capable of self-replicating, meaning divide. And it can also fuse. Which other organelle was capable of replicating? Anybody remembers? The peroxisomes. Okay, so let's look at the structure. It looks like a little sausage. The size can vary, and the number can vary. If you need more ATP, you need more mitochondria. Less ATP, less mitochondria. There are shorter and longer versions of mitochondria. And let's look at the parts. First of all, it's one of the membranous organelles. So again, what does that mean? It's made out of phospholipid bilayer, right? Just like the plasma membrane. Let's look at the parts. This is another organelle that has two plasma membranes. Which was the other one that had two plasma membranes? The nucleus, right? So this would be the outer membrane. Then there is an inner membrane, but the inner membrane makes these finger-like projections, these loops, in and out, in and out. That's what we call cristae, which increases the surface area inside the mitochondria. Now, between the outer membrane and the inner membrane, there is also an intramembranous space. So what is the inner membrane for? This is the enzymes for the oxidative phosphorylation sits. So let's call it like some kind of biochemistry is going on there. You didn't learn this yet in biochemistry, right? Okay. So just remember enzymes are sitting there. Okay? And the lumen inside is called the matrix, okay, which is basically between the cristae. And there are not another set of enzymes are sitting there for beta oxidation and Krebs cycle. So again, just remember, heavy biochemistry is going on the cristae and in the lumen. Okay, so what is really interesting that this guy has its own DNA and it's capable of making 13 protein in-house. What does that mean? Most proteins are coming from where? From outside, right? Nucleus, ER, Golgi, vesicle, protein comes over to the mitochondria. That would be the normal, usual way. But this guy actually is capable of making his own 13 in-house made proteins. So it has its own protein machinery, starting with the DNA. And the little DNA looks like a circular chromosome, like this. Okay, so because of this, lots of people there's two interesting things basically about mitochondria. So this is one of them. And because of this characteristic that it can make its own protein, and because it's self-replicating, people thought that maybe this was a bacteria originally and became symbiosis with us in the human beings. I personally don't believe that, but some people theorize that. And now you understand why we call this semi-autonomous, because it is making his own protein, which comes with its own blessing and cursing. If there is any mutation in this circular chromosome, which is located here on the image, you see, then there are not enough proteins made. And this is what we're gonna call mitochondrial disease. Okay, and this is where the three-parent story comes in that I'm going to describe to you in a minute as to introduce you to a little clinical connection. What is this guy doing all day long? Everybody knows it is synthesizing ATP with aerobic respiration. What does it mean? It needs oxygen. Okay? What you also need to, and actually it was in the movie what I showed you, and I do recommend that now that we finished all cell biology, you watch the movie again. Because now you're going to recognize a lot of things that you wouldn't, because in the movie you can actually see that the mitochondria is mobile and the microtubules are guiding it, some cytoskeletons are guiding it to move to one place to the next. Where is he moving? To the place where you need more ATP, okay? And again, here comes the self-replicating feature. Their number can increase by division, just like a bacteria, if more ATP is needed in another location. And their number can decrease by fusion. So this is the way you're gonna make long mitochondria. As I said, there are short and long ones. And they can be also autophage, meaning destruction. Yes, located in aerobic cells, and especially all of those cells that are extremely active. Like, for example, somebody counted in the liver cells 2,000. Okay? So let's talk about the clinical connection, the clinical three-parent story. And basically, there are children who are born with mutation in later. Mitochondria, what? You mean you cannot hear me? What? I cannot hear you. Even closer. Put it in my mouth. Okay, good. Is it better now? Okay. I thought it was close enough. Okay. But anyway, so if there is a mutation in the gene of the mitochondria, that little circular DNA, what I just showed you, then the child is gonna be born sick. So what they were trying, and here's the link, you can read about it. In the UK, they're trying to use another mother, a second mother, so to speak, who is donating mitochondrial DNA to fix the problem. So this is where the terminology, the three parents, come from, because the child is gonna have two mothers and one father. I think it's pushing it, but I think that's the way everything says if you make these weird big names. But anyway, why not take it from a male, guys? Why from another woman? Yes? Yeah. You know why? Better I don't ask any questions, because your mask is on, I don't hear anything. Mitochondria is always coming from the females. Okay, it never comes from the males. Okay. All right. So let's a couple of images here. So this is the example. Again, guys, remember, please start with the title, so you know what this title is all about. This is talking to you about the short mitochondria. Okay, the smaller ones, and light microscopic image with a special stain. They stain that enzyme, mitochondrial enzyme, with a blue dye. And you can see this has less mitochondria, this has more mitochondria, this has a lot of mitochondria. And this is the way your skeletal muscle actually looks like, that some of the skeletal muscle fibers are loaded with mitochondria, some of them very little mitochondria, and some of them is between. So therefore, the loaded with mitochondria is the one that you're gonna learn later. They're gonna be the aerobic fibers. They are the one that needs oxygen, and you use it when you are running. But that's another lecture altogether, okay? More ATP, more oxygen, you get on the treadmill. That's when those fibers become active, okay? Because muscles are usually a combination of these three kinds of muscles, and the physiology people will talk to you all about this. Now, if you look at in electron microscopic image, which this is a skeletal muscle, you can see that between the myofibrils, the parallelly organized mitochondria. Very easy to recognize because of the sausage shape and the cristae. Okay? So, how many cells do you see? Again, we have to get used to the look of the light microscope, shifting to electron microscope. How many cells do I see in cross-section on the light microscope image? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 2 half. 10, 12 cells I can see on light microscope. And how many cells do I see on the electron microscope? A corner of 1. Okay, not even 1, just a little piece. Okay, please get used to this, that electron microscope zooms in so ridiculously high. Okay, so next image is showing you an example of the long mitochondria. And usually salivary glands are famous for. These mitochondria actually sitting in this duct and in these basal striations that we call interdigitations of the plasma membrane. What does it mean? That the plasma membrane at the basal membrane here, basal plasma membrane, is making loops in and out. That's what we call interdigitation. Watch the word digit. You guys know what digit means? Finger. That's where the digital age comes from, because all we do is typing with our fingers. Okay? So look at it. So at the base of plasma membrane of this, what kind of cells do you see here? Columnar epithelial cells. Here it is. Look at it. This is the nucleus. You can see round and oval mixture. And this is where the secretion is going. So anyway, do you see the pink, white, pink, white, pink, white? Wherever it's pink, that's where the mitochondria is. You remember I told you mitochondria is pink, usually? Then wherever is white, that's where the plasma membrane is looping, because plasma membrane doesn't stain with hematoxin. Okay? So let's do it. Pink, white, pink, white, pink, white. Mitochondria, plasma membrane. Mitochondria, plasma membrane. One by one, one by one. Some people call this striation because it looks like a pink, white zebra. Okay? Now if you see the zoom zoom sign? Magnify this in electron microscopy. Now you're gonna see black, white, black, white, black, white, black, white. And the same thing, the black is the, and really long is the mitochondria. The white is the loop of the plasma membrane in and out, interdigitation. So black, white, black, white, black, white. Okay? And parallelly, exactly the same image, zoomed in. And again, how many cells do you see in this duct as we are in the light microscope? Again, we have to count the nuclei, right? 1, 2, maybe 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Columnar epithelial cells, okay? How many cells do you see in the electron microscope? 1, very good. 1 corner of 1 cell, just like this, you see that? Just something teeny tiny out of 1 cell, okay? So, let me see what happens now. This is where we finish the cell biology lecture, and I'm gonna do attendance. First for yesterday, and then for today, okay? Go away. Okay. Get ready, please, with your telephone. And please help me that I choose the right location. Wait a minute, this one we have to do first, right? Okay, so I'm clicking on that. Yes, I have to say no here, right? Okay. Are you guys ready? 1, 2, 3. What? What? I think the only reason is that you need more ATP, number one. Number two is, I think that you can see it especially with the duct. You know, you have this long territory just for the mitochondria itself. Not every cell can accommodate a long mitochondria. So it's the size of the cell itself also helps. Okay, guys, ready for, let me save it. You're welcome. So let me save it. No, I'm not gonna repeat it because we don't have time for that. Please email me if there is a drama. Okay, and now we are doing this morning, okay? Can you guys get ready? So it's this lecture now. This is it, right? Monday, yeah. I have to say no somewhere here, okay? No. Generate attendance, okay? If you have any problem, please come closer to the, that's really weird. It didn't become, here we go. QR code was late. Is it working? Okay, thank you. So anybody has problem, please please email me, okay? And I fix it for you. And when you wait a minute. Okay, you guys are in one group right now because on week 1 you guys were in 3 groups. So, okay, let me save it. Yes. What? As I said, please email me, okay? So I will fix it for you. Okay, so let's move on to, we're gonna do a little bit of connective tissues. We already gave you a little bit of introduction, you know, like a week ago or 2 weeks ago. And connective tissues is very simple between you and me. All you have to remember is boom and boom and boom, okay? Number 1. So let's do the boom, boom, boom. Connective tissues are coming up. Enable editing, very good. And then I remember to turn the pointer. Good. So we're gonna start talking connective tissue. The name already tells you that it's gonna be between tissues. Okay, so you can anticipate that it's gonna be right away next to the epithelium. This is what we call underlying connective tissue. That's one thing. The other thing is between tissues. And you're gonna learn these big words. It's located in the lamina propria in the mucosa. And it is located in the stroma of organs. Stroma is the supporting tissue of any tissue, and that's connective tissue. It supports. What does it support? The parenchyma. What is the parenchyma? That part of the organ that is actually doing the function of that particular organ. So get used to these 2 terminology. Stroma is supporting tissue, which is connective tissue, and the functioning tissue is the parenchyma. Also, one more terminology here, that you're gonna see a lot of cells right now, and whenever they cell blast, those are those cells that actively synthesizing the connective tissue itself. When you see site, usually those cells are retired and they are maintaining the connective tissue. Now this is true for all cells except the fibroblast. We're always going to use the word fibroblast. Okay? I don't know why that happened in literature, but that's just the way it is. These are your objective structure to compare to the epithelium. And we already a little bit talked about this, I believe, but one of the major things I want you to remember that connective tissue has only few cells, so no cellularity. What do I mean by that? Epithelium was epithelial cells on top of each other, no break, right? To make sure that nothing is leaking through. Connective tissue is a loose tissue, okay? The cells are scattered far away from each other, therefore there are no cell junctions between them because there is no need, and there's a huge extracellular fluid all around them, and that's what we call ground substance. I mean, histology people call it ground substance. It's really regular extracellular fluid. And this is where protein fibers are also going to be located. And of course, as a result, these cells don't have polarity like epithelial cells because they are just sitting in the middle of collagen. Okay, now what connective tissues in general have a very good rich blood supply, except the tendon. It has nerves. It quickly regenerates, and you know, actually epithelium also quickly regenerates, right? And finally, of course, you already know this from Dr. Zafar, when they are active, you can see large euchromatic nucleus with very prominent nucleoli. So here is an image already for you, the very tight millions of cells of a stratified epithelium, right? No break, and here these flat nuclei buried in pink collagen are, and the blue cells are far away from each other. That's the typical connective tissue. Okay, so this is a table for you, just what I just did. Okay? So the only thing, again, and I already did this a few times, so I hope you're going to remember, connective tissue is famous for extracellular protein fibers. You remember epithelial cells were famous for intracellular cytoskeleton. Now, cytoskeleton can show up in connective tissue cells as well, but I'm just reminding you the two types of proteins again, okay? Okay, so here comes the boom, 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 guys, okay? And you have the living part of the tissue where the cells are, okay? The number one most important, most common cell is the fibroblast. Then comes white blood cells or leukocytes for defense. And these are the most famous one: macrophage, mast cell, leukocytes. Famous adipocyte. Adipocyte or fat can show up anywhere, unfortunately. We cannot get rid of it. And yes, there are some mesenheimer undifferentiated cells that people use shows up in embryo, but it's also can be stem cell in an adult. We talk about it later. Okay, so these are your living cells. So what is the fibroblast is doing all day long, guys? This. The fibroblast is creating the connective tissue, the non-living part. You see, I put it here for you, synthesized by fibroblast. What is that? The BOOM2 and the BOOM3. The BOOM2 is the protein fiber ropes, collagen, elastin, and reticulin, that is actually type 3 collagen. So these are your large extracellular proteins, fibrous proteins. And on the other side, the fibroblast also synthesizes into the extracellular fluid the GAGs, which is called glycosaminoglycans. The most important example is the hyaluronate. and proteoglycans. These guys all gonna go into the ground substance. So it's not just pure water, and these molecules are going to make the ground substance jello-like. Now that we got this straight, the three major components, let's look at it on this image, okay? So we have to find the fibroblast, which is a flat cell buried usually in collagen. Here it is. That would be a good collagen. The defense cells are usually round. Here's another nucleus, a round one. Let's look at the protein ropes. The thick, strongest fibers are the collagen. The thin pink fibers are the elastin protein fibers. Reticulin is not stained on this image, just on the side. With hematoxin eosin, probably doctors have already told you, collagen is beautifully visible because it's a large protein. Elastin and reticulin both needs a special stain to make it thicker so you can see them. Okay? And finally, in light microscope, in hematoxylin eosin, you will not be able to see the GAGs and the PGs. Okay? They also need special stain. Okay? So basically that's all what we're gonna do, going through one by one the cells, the fibers, and a little bit of the components of the ground substance. So here we go. Classification of connective tissue cell types. So there are two kinds of cells in the connective tissue. There are one that is always there, okay? They are permanent resident. Did I make a mistake here? They are not, okay, I have to change that, guys. I'm gonna change that. They are not immigrant cells. They are permanent resident. And the fibroblast, adipocyte, mast cell, and macrophage are always in the connective tissue, okay? And here are their functions. Fibroblasts we already know. Adipocytes store fat for energy. Mast cells is trigger inflammation. And macrophage is usually phagocyte, remove foreign material and damage cells. Now, if you talk about the white blood cells, that was, here, the white blood cells, wait a minute, here, leukocytes, white blood cells, then these guys are the one that are immigrant cells. That's why I have to immigrant remove from here. I don't know how I made that mistake, but it's, after I finish this lecture, I will fix it and upload it again, okay? So they are actually circulating in the bloodstream, and if they receive a signal that there is an infection somewhere in the body, then these white blood cells can exit the bloodstream and go into the connective tissue. Okay, so let's talk about fibroblasts first. As you can see, this is the way you're gonna see fibroblasts in a light microscope. Elongated cell, elongated nucleus, and here is the thick collagen fibers between. So boom, boom, and the white nothing is the ground substance, boom. Okay, now let's look at this cell in electron microscopy, and let me show you. This is your cell from here to here, to here, to here, okay? And this is the flat nucleus. Okay, so you see how many cells can you see in light microscope? 10, right? How many cells do you see in the electron microscope? Half, very good, half, okay? And you can see a little bit of the organelles, mitochondria, endoplasmic reticulum, Golgi. Buried in collagen. So look what I'm showing you. Do you see this zebra sitting here? That's collagen fibers in longitudinal section. If you look at collagen fibers on electron microscopy, they have striations on them. And what about here? Collagen fibers in cross-section. And I want to tell you a secret. Everything you see in the body in cross-section is a circle, everything. The real structures that are significant or characteristics are usually show up in the longitudinal view. Remember that, okay? So here we go. I'm gonna remove this slide, okay? So we're gonna continue and talk about the Mesenheimer cells. Okay, so the Mesenheimer cells are originated from the mesoderm. Did you guys learn ectoderm, mesoderm, and okay, wonderful. Okay, so anyway, you have in embryology you're gonna start with the 3 layers of a little human being. Start with 3 layers. The middle is the mesoderm, and that's where the mesenchyme is coming from, which is the undifferentiated Mesenheimer cells, and all connective tissue cells are coming from here as the baby is developing, okay? And some of them in adult become stem cells. Okay, so this image, it looks overwhelming, but it's really not. This is your undifferentiated Mesenheimer cell, as I said, in the mesoderm, and eventually it's gonna become connective tissue cells. These are all connective tissue cells, guys. So far we covered the fibroblast. Even adipocyte is another connective tissue cell. Then these are the cartilage cells, and these are the bone cells. And very interestingly, the smooth muscle in blood vessels and mesothelium, which are not connective tissue, also developed from mesenchyme. Now, how does this look on a light microscope? So here you have to find again, boom, 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 okay? The Mesenheimer cell is kind of like a star shape or elongated, depends on how you cut it, but it's really a star-shaped cell. So if you cut it this way, you see the star. If you cut it that way, you don't see the star. And very fine immature collagen fibers, and the white nothing is the ground substance. So boom, boom, and boom, done, okay? So let's move to the next slide, and we're gonna focus on the cells of defense. So these cells, or leukocytes, are originating from the red bone marrow, guys, not from the mesenchyme, not from the mesoderm. They are coming from the hematopoietic stem cells. So if you look all these around, by the end of this year, you're gonna know all these cells, okay? But most of them are blood cells, and they are in the blood, okay? And then some of them exit, and they have their own different functions. Only one I picked out for you, that the monocyte, when it's basically can have 3 locations. Actually, all of them can have 3 locations. Start in the bone marrow, exit the bone marrow and go into the blood, and now it circulates in the blood, and then when it goes into the tissue where it's gonna finally do its job, that's location number 3. And the monocyte is the one that people use when it's in the blood. When it enters the connective tissue, you're gonna call it macrophage. Probably you already heard this name before. The old name for this guy is histiocyte. Okay, now this image is showing you how a macrophage, which literally means the big eater, or histiocyte, how it looks like in light microscope and electron microscope. Okay? A function, innate immunity. What does it mean? This is what we are born with. This guy is a general immune reaction to invader. If there is a bacterial virus infection, it's immediately response. It's not a specific response. That's what you call innate immunity. Okay, this is the way macrophage looks like large round cells, and a roundish, depends on how you cut it, or bean-shaped nucleus. They are, you see, macro. Again, watch what you are saying, because the names already tells you a lot of things. Macro says large. Phage, it's eating. So 21 micron large cell, and again, I would like to remind you that the purpose of these numbers, when we put micron here, micron, then nanometer, blah, 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 it's more like the comparison. And what you want to compare everything with is really the erythrocyte. The erythrocyte is the best to use as a ruler for your mind. That's going to be the 7 micron, which is also the size of the smallest capillary where the red blood cell can go through. So after that, if it's smaller or bigger, you can say, wow, this is really small because it's smaller than the red blood cell. Wow, this is really big because it's 3 times bigger than the red blood cell. So that's what I want you to think, not like I'm going to test you in every single this number, but I want you to kind of have a feeling of the size. Okay, it's going to be activated when there is an infection or a tissue damage in the body. And this is when it moves by this pseudopodia. Remember I showed you the movie yesterday? Who is helping the movement of the pseudopodia? The actin filaments. Actin filaments are the muscle of all cells, muscles and non-muscle cell. This is a non-muscle cell, like what I said, or I called this non-contractile cell. Okay? So these guys move around with actin growing this direction, that direction. Okay, now he's gonna go phagocytosis. It's like with the pseudopodia, nyam, and takes it in, swallow it. Okay? Then lysosome is going to digest the antigen or the bacteria. Then the antigen is gonna be present to the lymphocytes, and this is when the adaptive immunity wakes up. So what did the macrophage just did? The innate immunity woke up the adaptive immunity. Adaptive immunity now is going to be specific for that particular bacteria. Okay? All right, so that what you see on the electron, again, you see light microscope, how many cells I see? 5, and many other types of cells. In the electron microscope, 1. You see? This is the kind of bean-shaped nucleus, and these, they fooled this macrophage. They fed him with latex particles. And the latex particles, these white circles, and they made this image early after feeding, so only 1 lysosome is showed up on the image, according to the people who made this electron microscopic image. Now this is the mast cell, guys, or mastocyte, and that's what you see on this image. The mast cells are very similar to basophil granulocytes. They are small, 15 micron. Do you see that? This is double than the red blood cell. And it has 80 large blue granules that has histamine in it, and it's bilobed. It has 2 lobes. And now, this was the basophil granulocyte. Now, let's look at how the mast cell looks like. Looks identical, except you're going to see 1,000 tiny blue granules with histamine. Okay? When I went to medical school, then they still thought that this 2 cell is the same. And then, again, the electron microscope really helped to discover that the basophil granulocyte has a bilobe nucleus. And here it is, look, this is the mast cell that has 1 round nucleus. This guy has, the basophil granulocyte has 80 large granules with histamine, and the mast cell has 1,000 tiny blue granules with histamine. Okay? But both of them are involved in the same functions, immediate hypersensitivity, or you may know this word anaphylaxis, allergic reaction, in asthma, urticaria, or in anaphylactic shocks. Okay? Now this is all the cells that you can see in the connective tissue. You can see again the fibers left and right, right? The ground substance is the blue nothing, and all kinds of cells. Okay? You don't need to all of these cells up till now, the only one that I covered. Okay? The blood cells you're gonna learn later. Okay, let's talk about the fibers. So what is the functions of the connective tissue? How did I get here? Okay. I thought this was supposed to be at the beginning, but I don't know. Okay, so what does the connective tissue really does in your body? It's mechanical support for epithelium, for nerve, for muscle, and blood vessels. Okay, and you're gonna learn all these big names very soon. You remember the epithelium is avascular, then comes the connective tissue, and the connective tissue has the capillary that keeps the epithelium alive. Okay? Way of communication and transfers among different tissues. So basically, as molecules exit the capillaries, bloodstream, they will travel through the ground substance to reach a cell to bring the signal. That's what we mean. This is the place of inflammation. Blood cells, we just talked about it, the white blood cells are gonna make the big show here, especially with the presence of and secretion of histamine. The transport of oxygen and nutrient is also gonna go through the ground substance. And remember that, guys, that scar tissue is made out of collagen. And I have to change this cell, and I already did, but I uploaded the wrong version of this lecture. The fibroblast actually becomes or differentiate into myofibroblast, and that is going to be involved in scar tissue development. Adipose tissue, everybody knows, energy storage. And if you didn't know that, bone marrow is a specialized connective tissue to make blood cells that we call hematopoiesis. Now let's talk about we covered a few cells, right? Fibroblast is number one, the most abundant, most important. And I covered 3 immune cells for you. Okay, so now we're gonna move to the boom 2, which is the collagen fibers. These are the large extracellular fibrous protein, and I call them ropes because they're strong like a rope. Have you seen a big rope when they are tying a cruise ship to the port? If you didn't, then watch Pirates of the Caribbean because it's all there. Okay, and these guys are white protein fibers. What does it mean? It means that they are really white. In real life, they are really white. In the microscope, you're gonna see them pink because we stain them. So where do you see collagen when you guys looking at each other? The white of your eye. That's collagen, guys. So this is really white, okay? And when you go into the growth lab, and you're gonna see, you open the muscles or the organ and everything, you're gonna see collagen everywhere, and they are always white. How about the fat? How is the fat usually looks like? More like yellowish, right? So remember that when you go into the growth lab. Now, I also wanted to tell you that when you buy this dessert, gelatin, it is actually denatured collagen. So all you have to do is just cook the bone. Bones are loaded with collagen. We're gonna cover that in the next block. You boil the bones for like 8 hours, collagen comes out, you cool it down, and it becomes like this jello. That's the way they make the gelatin. All you have to do, just put a little bit of strawberry on the top, and done. Yum, yum. Okay, now this slide is definitely not for memorization. It's a biochemical image. But I want you to see 2 things. Number one, this is the active fibroblast that is synthesizing what? The little monomers of the collagen. So here is your ER, the Golgi, the exocytotic vesicles. You see, these are the little pieces of the collagen that is spit out, okay? And the big, large collagen fibers actually assembles outside in the extracellular fluid. The big ones not gonna fit in there, right? So you just make the little pieces like a Lego, right? And you put it together outside. And again, this is your ER, this is the Golgi. And this is where, you remember, this is where you need vitamin C at this step. Then here is your little monomer, exocytosis, and this is the little piece that comes out. Then you start assembling bigger little ropes. And here's the big one. So people usually use the terminology as, you see, monomer, fibril, fiber. Sometimes filament. So when you say filament or fibril, you are referring to a smaller, thinner fiber. When you say fiber, then it's usually a big one. So even the terminology helps you. But this is where we come to the thing that, okay, I will talk to you about later. But anyway, you guys remember this, my medical school? I already showed you this, so forget it. So let's look at the collagen fibers now. Same image I showed you earlier, but now we are focused on the collagen. In hematoxin, I was in the white fibers are pink. And you see how nicely huge, parallelly organized on this image. Then you zoom in, and then you see the striation with electron microscopy that I already showed you on a previous image. And then here's the assembly of the pieces to make a tropocollagen triple helix monomer that is eventually is going to make the huge collagen fiber. Okay? Okay, so this is an important slide. So look at, this is from Pirates of the Caribbean. See the ropes? And it tells you that the striation is every 68 nanometer. Did you see nanometer? Very tiny distance. So that's why you don't see it on light microscope. Even though I did see it before on light microscope, it depends how good the slide is. Okay, what is the collagen doing all day long? Resist pulling. So if anybody is pinching you, for example, on your hand too much, basically the collagen is pulling you back. So nobody can tear you apart. You understand? Collagen is pulling back, resist the pulling force. Okay, so remember, type 1 collagen is the #1 and most abundant. So most of the time, that's what we're going to see in the lab. Type 2 collagen is usually in cartilage. Type 3 collagen, which I already mentioned, reticulin fibrils, are always in those organs that housing cells upon cells upon cells. And the collagen type 3 is going to be the little place for them to sit down, like you need a chair to sit down. Imagine you are a cell, your chair is the collagen type 3. You have to be able to have your own little place. Collagen type 4 is in the basement membrane, and collagen type 7 anchors the basement membrane of the epithelium to the connective tissue below. Okay? You remember where we are right now? Right where the hemidesmosome is sitting. Okay? So here we go. So you remember, this is your hemidesmosome 1, hemidesmosome 2. So on the top, this is basically an epithelial cell. Here you see 1, 2, 3 epithelial cell. This one is basically just one, magnified. And here comes type 4 collagen in the basement membrane 1. And this is what I just said. And the type 7 collagen loops and anchors the basement membrane to the deeper type 1 and type 3 collagen. Okay, so now let me just look at the time. So this is the light microscopic image of the basement membrane with a special stain. You remember, it does not stain with hematoxylin eosin, so you need a special stain, PUSH, which is basically means a dye that stains sugar, anything glyco. Okay, and how does the basement membrane looks like in electron microscope? So again, this is your cell, epithelial cell. That's a hemidesmosome, HD. And the three layers are lamina lucida, lamina densa, and lamina fibroreticularis. When you really, really zoom in, it becomes three layers. But again, watch what you are saying. The lumina lucida means Lucifer, the devil. What does Lucifer means, guys? The angel of light. So what they're really saying that the light layer, then lamina densa, which is black, LD, dense layer, it's not that difficult, light layer, dense layer, and do you see how the fibers are crisscrossing with each other here? That's your fibroreticularis, because reticulum means networking and branching. Okay, so that's the way the whole thing looks like. So this is the same image, just next to each other so you can connect. Okay, so this is where we're gonna continue after 10 minutes break.